Lysine is an I+ or minus amino acid with the chemical formula HO2CCH, NH2, CH2, 4NH2. It is an essential amino acid for humans. Lysine's codons are AAA and AAG. Lysine is a base, as are arginine and histidine. The I-micron amino group often participates in hydrogen bonding and as a general base in catalysis is attached to the fifth carbon beginning from the I+ or minus carbon, which is attached to the carboxyl group. Common post-translational modifications include methylation of the I-micron amino group, giving methyl, dimethyl, and trimethylysine. The latter occurs in carmodulin. Other post-translational modifications at lysine residues include acetylation, sumoylation, and ubiquitination. Collagen contains hydroxylysine, which is derived from lysine by lysyl hydroxylase. O-glycosylation of hydroxylysine residues in the endoplasmic reticulum or Golgi apparatus is used to mark certain proteins for secretion from the cell. Biosynthesis: as an essential amino acid, lysine is not synthesized in animals, hence it must be ingested as lysine or lysine-containing proteins. In plants and bacteria, it is synthesized from aspartic acid. L-aspartate is first converted to L-aspartyl-4-phosphate by aspartate kinase. ATP is needed as an energy source for this step. I-squared-aspartate semialdehyde dehydrogenase converts this into I-squared-aspartyl-4-semialdehyde. Energy from NADPH is used in this conversion. Dihydroadipicolinate synthase adds a pyruvate group to the I-squared-aspartyl-4-semialdehyde, and two water molecules are removed. This causes cyclization and gives rise to 2,3-dihydroadipicolinate. This product is reduced to 2,3,4,5-tetrahydroadipicolinate 2,3,4,5-tetrahydropyridine 2,6-dicarboxylate, by dihydroadipicolinate reductase. This reaction consumes a NADPH molecule. Tetrahydroadipicolinate N-acetyltransferase opens this ring and gives rise to N-succinyl L2-amino 6-oxyheptanedeonate. Two water molecules and one acyl-CoA enzyme are used in this reaction. N-succinyl L2-amino 6-oxyheptanedeonate is converted into N-succinyl LL2, 6-diaminheptanedeonate. This reaction is catalyzed by the enzyme succinyl diaminopymalate aminotransferase. A glutamic acid molecule is used in this reaction and an oxoacid is produced as a byproduct. N-succinyl LL2, 6-diaminheptanedeonate is converted into LL2, 6-diaminheptanedeonate by succinyl diaminopymalate desuccinylase. A water molecule is consumed in this reaction and a succinate is produced a byproduct. LL2, 6-diaminheptanedeonate is converted by diaminopymalate epimerase into meso-2, 6-diaminoheptanedeonate. Finally, meso-2, 6-diaminoheptanedeonate is converted into L-lysine by diaminopymalate decarboxylase. Enzymes involved in this biosynthesis include aspartate kinase, I-squared-aspartate semialdehyde dehydrogenase, dihydropicolinate synthase, I1-piperidine 2, 6-dicarboxylate dehydrogenase, N-succinyl 2-amino 6-catepymalate synthase, succinyl diaminopymalate aminotransferase, succinyl diaminopymalate desuccinylase, diaminopymalate epimerase, diaminopymalate decarboxylase. It is worthwhile to note, however, that in fungi, euglenoids and some prokaryotes lysine is synthesized via the alpha-amino adipate pathway. Metabolism: lysine is metabolized in mammals to give acetyl-CoA, via an initial transamination with I+ or minus ketoglutarate. The bacterial degradation of lysine yields cadaverine by decarboxylation. Allicine is a derivative of lysine, used in the production of elastin and collagen. It is produced by the actions of the enzyme lysyl oxidase on lysine in the extracellular matrix and is essential in the cross-link formation that stabilizes collagen and elastin. Synthesis: synthetic, racemic lysine has long been known. A practical synthesis starts from caprolactam. Industrially, L-lysine is usually manufactured by a fermentation process using Corynebacterium glutamicum. Production exceeds 600,000 tons a year. Dietary sources: The nutritional requirement per day, in milligrams of lysine per kilogram of body weight, is infants 103, children 64, older children 60 to 44, adults 12. For a 70 kg adult, 12 milligrams of lysine per kilogram of body weight is 0.84 grams of lysine. Note that recommendations were subsequently revised upwards, for example 30 milligram per kilogram for adults. Good sources of lysine are high-protein foods such as eggs, meat, soy, beans and peas, cheese, and certain fish. Lysine is the limiting amino acid in most cereal grains, but is plentiful in most pulses. Consequently, meals that combine cereal grains and legumes, such as the Indian dal with rice, Middle Eastern hummus, full medams, falafel with pita bread, the Mexican beans with rice or tortilla have arisen to provide complete protein in diets that are, by choice or by necessity, vegetarian. A food is considered to have sufficient lysine if it is at least 51 mg of lysine per gram of protein. Foods containing significant proportions of lysine include, properties. L-lysine is a necessary building block for all protein in the body. L-lysine plays a major role in calcium absorption, building muscle protein, recovering from surgery or sports injuries, and the body's production of hormones, enzymes, and antibodies. Modifications: lysine can be modified through acetylation, methylation, ubiquitination, sumoylation, nedylation, biotinylation, pupylation, and carboxylation, which tends to modify the function of the protein of which the modified lysine residue, S, are a part. Clinical significance: some studies have found that lysine may be beneficial for those with herpes simplex infections. One small randomized, controlled study found that it reduced outbreaks by 2.4 per year. Other studies found no benefit. Lysine has not been approved by the FDA for herpes simplex suppression. Lysine has anxiolytic action through its effects on serotonin receptors in the intestinal tract, and is also hypothesized to reduce anxiety through serotonin regulation in the amygdala. One study on rats showed that overstimulation of the 5-HT4 receptors in the gut are associated with anxiety-induced intestinal pathology. Lysine, acting as a serotonin antagonist and therefore reducing the overactivity of these receptors, reduced signs of anxiety and anxiety-induced diarrhea in the sample population. Another study showed that lysine deficiency leads to a pathological increase in serotonin in the amygdala, a brain structure that is involved in emotional regulation and the stress response. Human studies have also shown correlations between reduced lysine intake and anxiety. A population-based study in Syria included 93 families whose diet is primarily grain-based and therefore likely to be deficient in lysine. Fortification of grains with lysine was shown to reduce markers of anxiety, including cortisol levels. Smyriga and colleagues concluded that anxiety reduction from lysine occurs through mechanism of serotonin alterations in the central amygdala. Older primary research reports hypothesized lysine to reduce anxiety through the potentiation of benzodiazepine receptors. There are lysine conjugates that show promise in the treatment of cancer, by causing cancerous cells to destroy themselves when the drug is combined with the use of phototherapy, while leaving non-cancerous cells unharmed. While chemically insignificant to lysine itself, it is worth noting that lysine is attached to dextroamphetamine to form the prodrug lisdexamphetamine. In the gastrointestinal tract, the lysine molecule is cleaved from the dextroamphetamine, thereby making oral administration necessary. Lysine deficiency causes immunodeficiency in chickens. One cause of relative lysine deficiency is cystinuria, where there is impaired hepatic resorption of basic, or positively charged amino acids, including lysine. The accompanying urinary cystine results because the same deficient amino acid transporter is normally present in the kidney as well. Limited studies suggest that a high lysine diet or L-lysine monochloride supplements may have a moderating effect on blood pressure and the incidence of stroke. Use of lysine in animal feed. Lysine production for animal feed is a major global industry, reaching in 2009 almost 700,000 tons for a market value of over $1.22 billion. Lysine is an important additive to animal feed because it is a limiting amino acid when optimizing the growth of certain animals such as pigs and chickens for the production of meat. Lysine supplementation allows for the use of lower-cost plant protein while maintaining high growth rates, and limiting the pollution from nitrogen excretion. In turn, however, phosphate pollution is a major environmental cost when corn is used as feed for poultry and swine. Lysine is industrially produced by microbial fermentation, from a base mainly of sugar. Genetic engineering research is actively pursuing bacterial strains to improve the efficiency of production and allow lysine to be made from other substrates. In popular culture, the 1993 film Jurassic Park features dinosaurs that were genetically altered so that they could not produce lysine. This was known as the Lysine Contingency, and was supposed to prevent the cloned dinosaurs from surviving outside the park, forcing them to be dependent on lysine supplements provided by the park's veterinary staff. In reality, most vertebrates cannot produce lysine. In 1996, lysine became the focus of a price-fixing case, the largest in United States history. The Archer-Daniels-Midland Company paid a fine of $100 million, and 3 of its executives were convicted and served prison time. Also found guilty in the price-fixing case were 2 Japanese firms and a South Korean firm. Secret video recordings of the conspirators fixing lysine's price can be found online or by requesting the video from the U.S. Department of Justice, Antitrust Division. This case served as the basis of the movie The Informant and a book of the same title. See also, acetyllysine, deamination, lysine price-fixing conspiracy, methyllysine, saccharopin. References. Sources. Much of the information in this article has been translated from German Wikipedia. Lide, Dr. ed. CRC Handbook of Chemistry and Physics. Boca Raton, Florida, CRC Press. ISBN A0-8493-0483-0A.